Hi, today I wanted to expand a little bit on Objective 3 in your Chapter 15 lecture notes. I had thought this would be a fun way to give you some more clinical experience since I haven't had the chance to do any hands-on experiments with you guys due to the pandemic. So some of this information, it might even help you with your extra credit research paper, depending on which of the three topics you choose to work on. So I'm going to go ahead and jump right in. You probably have already looked over the notes for Chapter 15. They're up on Canvas, and I'm going to go ahead and skip over to the objective where you are asked to learn about blood, the composition of blood, and I'm going to spend most of our time on the formed elements, talking about white blood cells. So your lecture notes focus here on the whole blood sample, and then we'll talk about the formed elements. So let me first talk about a very common clinical practice. It's called venipuncture, and you get the patient's blood sample, and you carefully transfer it to a tube like you see here. And there are many different tests that require different preservatives, or maybe none at all, in order to evaluate the patient's sample. One of the most common laboratory procedures is to determine the hematocrit. This is going to tell you about the patient's hydration status and overall general health. In order to evaluate this blood sample, it must be spun down in a centrifuge first using a gentle setting so we don't rupture the red blood cells. That would result in a pink sample, and you would have to trash that sample if that happened. The results here, you can see there's separation, 3 different phases. And on top, the yellow straw-colored fluid plasma. It's about 55% of the entire sample volume, and that's going to be on the top. And then below that, if you look really closely, is what's called the buffy layer. It's this small, very thin layer. It's only about 1% of the sample's volume, and it really does look like someone poured talcum powder in your sample. That's where the white blood cells are found, and that's where I want to spend most of our time expanding upon Objective 3 with you guys, is talking about the white blood cells. It's the smallest part of this hematocrit, but the white blood cells have a lot of different immune function, and I wanted to talk a little bit about that, kind of help you guys with your COVID research papers, and also talk about this before we get into the respiratory system, which we all know the virus likes to use a protein to gain access to human cells that's found pretty commonly in the lungs. Down at the bottom of this test tube, you see the bright red blood cells. That's the bottom layer, and they're packed to the bottom when you spin the sample down. The red blood cells are about 45% of the total volume, and this is just a really common value, clinical value, that you'll get on most of your patients. And let's go ahead and talk a little bit more about the formed elements and focus on the white blood cells. The production of all formed elements found in a blood sample occurs in the bone marrow, and this process is called hematopoiesis. This process starts with a single stem cell, which, as you can see up here at the top, it's going to differentiate into 7 very different cell types, which includes the red blood cells, all the white blood cells, which are part of the immune system, like I mentioned, and platelets, which help in the clotting process. The platelets are over here on the far left. So this myeloid stem cell is going to be one branch of the 2 branches that you see. The hematopoietic stem cell up at the top can differentiate into anything, really. And that's part of the beauty of stem cell research. And here, for the formed elements, we have 2 lines, a myeloid stem line and a lymphoid stem cell line. So the myeloid stem cell line over here on the left is really the most diverse and forms the majority of the formed elements we would find in a blood smear. And then the lymphoid stem cell line over here on the far right is exclusive to the formation of just lymphocytes. And these formed elements along here at the bottom are the ones I want to focus on today. So when we get to our objective slide, we're going to see that We're going to talk all about the function of the formed elements, and this is a little different than what I normally do with most of my physiology classes. So I just wanted to do this a little bit differently for you guys. This is something that comes up a lot in your profession, in your future profession, but we don't spend a lot of time on the immune system or talking about the different white blood cells. So instead of reading from your slides like I normally do, I wanted to use some photos taken with 100x oil, magnified under the microscope, and go ahead and talk about these white blood cells. And you can see here this figure from your book in chapter 15, the comparison in size to the red blood cell. So the erythrocyte is small but much more numerous, and it lacks a nucleus. This is the only human cell that lacks a nucleus. And then the white blood cells do have nuclei, and they have some unique characteristics. So I want to go through each of these. The neutrophil, eosinophil, and basophil along the top row are all granular. They have little polka dots inside. So those granules add function to these cells. And then the lower row, those are agranular lymphocytes and monocytes. And the arrow between monocyte and macrophage, that is technically the same cell. So monocytes in circulation can invade tissues, and when they go into tissues, they become macrophage. So they do another step of differentiation, like we saw from stem cells into the mature cells that we see here in figure 15.6. Monocytes can differentiate even further and become macrophage. So I want to just go ahead and go through all of these white blood cells in a little more detail. And I can pull up your slide. This is a picture of a blood smear, and this is a good technique to learn how to do a blood smear. I'll include a college video on this technique so you can see how to do this if you're in the lab setting. And this is made from a single drop of whole blood. And you can see that the red blood cells are the most numerous, which is great since they carry the oxygen to all the tissues. And we can also appreciate one of the most commonly found white blood cells, which is the neutrophil. And there are also some platelets down here in the lower corner. They're clustered together. It's a very common looking slide if you were to be doing this in the lab under the microscope. It is very typical. And then we'll go ahead and talk about the most common granular site. This is the neutrophil. Let's take a look at him. Neutrophil being the most commonly seen white blood cell type. Another name for this is polymorphonuclear lymphocyte, or PMNs. PMNs can make up anywhere from 40 to 70% of all the white blood cells in mammals, and they are an essential part of the immune system. They're part of the innate immunity that we all have. We're born with it. You can see a uniquely shaped nucleus, characteristically divided into 3, 4, 5, maybe 6 different lobes. So this is a very unusual looking nucleus. It's not the solid round type of nucleus. So this is very typical for the neutrophil. The name neutrophil was given because of the staining characteristics, which give it kind of a neutral lavender color, not red and not quite blue. So neutrophils are neutral in the middle, and they can get recruited to the site of infections or injury within minutes, and they're the hallmark of acute inflammation. And since these white blood cells are highly mobile, neutrophils quickly congregate by following the chemical cytokines released from the site, and this is called chemotaxis. This in turn will amplify the localized inflammatory response. Neutrophils use 3 strategies for directly attacking microbial growth and invaders in our system. One is a process called phagocytosis, and that's a process of ingesting the foreign matter. Phago means to eat. They can also release soluble antimicrobial agents from their granules. And you can see the granules here in the cytoplasm. It looks maybe a little bit speckly under the microscope. And then their third method is they can set what are called neutrophilic extracellular traps, called nets for short. So these guys are the sentinels, the most common that you see on a blood smear, and they're the first on the scene during infection. Now, the lymphocytes are the second most commonly seen cell type on the smear under the microscope. This cell does not have any granules, so it's an agranular, and it has that nice round nucleus. So he's pretty common on the blood smear. I've zoomed in here a little bit more than what's on your copy of the lecture notes, so we can see this very large, dark staining nucleus, and there's very little cytoplasm here. This large, dense nucleus takes up about the entire ICF compartment, and they're pretty common, so they'll be anywhere from 20 to 45% of your white blood cell population if you were to do a cell count or a differential from a smear. There are two functionally distinct subsets of lymphocytes. They can correlate with their microscopic appearance. You have to have a pretty trained eye to really see some of these nuances, but I'm going to talk about some things that you guys might have heard about in other classes or in the news as far as immunity goes. The larger of the lymphocytes are commonly known as natural killer cells, and then the smaller subset of lymphocytes that make up a family of T cells and B cells. But you'd have to do some histology studies really to tell these three different subsets, the natural killer cells from a T cell and a B cell. But you can tell if you're careful and you're looking under the microscope based on size. So lymphocytes play an important role in the body's natural defense mechanisms. Natural killer cells can distinguish between virally infected cells and tumor cells from normal and uninfected cells just by recognizing the level of changes in surface proteins that belong to MHC class 1, which stands for major histocompatibility complex. If you were to donate an organ, they would blood type you and do some studies to see if you're a compatible donor. And major histocompatibility complex, or MHC1, is one of those things that they test for. Natural killer cells are activated in response to a family of cytokine chemicals called interferons. Activated natural killer cells will release cytotoxic, which means cell-killing, granules, which then can destroy any cells that have been altered by a foreign invader. T cells and B cells are the major cellular components of the adaptive immune response. So there's humoral and there's adaptive, or innate and adaptive. T cells are involved in cell-mediated immunity, whereas B cells are primarily responsible for the humoral immunity, which in this sense means relating to antibodies that circulate in the fluid portion of blood. The function of T cells and B cells is to recognize non-self antigens during a process known as antigen presentation. Once these cell types have identified an invader, these cells will generate highly specific responses tailored to eliminate the pathogen itself or a pathogen-infected cell. B cells respond to pathogens by producing large amounts of antibodies, which are able to neutralize pathogens like bacteria and viruses. Let's look at the monocyte now. This is a slightly better version of the photo in your lecture notes, so I wanted to discuss some of these unique characteristics of the monocyte using this image. It's classified as agranular, but in this photo we can get a better idea of what's on the interior, or in the ICF compartment, of a white blood cell type that looks like this. These little bubbles, or clearing zones in the ICF compartment, are vacuoles. Monocytes are the largest of all the white blood cells and are usually identified in stained blood smears by their large blue staining bilobed or U-shaped nucleus. Monocytes will circulate in the blood for about 1 to 3 days and then typically relocate by a process called diapedesis, and they move into the tissue. So they come out of circulation and into the tissues. When we talked about capillary beds in the previous lecture, we looked at fenestrated capillaries that would allow for movement into tissues from capillary beds into the tissues to help fight infection. These white blood cells may be a little bit more rare to find on a smear. They only make up about 4 to 8% of the total population. And during infection, monocytes can become localized within 8 to 12 hours. When monocytes come out of circulation and move into the infected tissues, they're going to undergo further differentiation, which makes them macrophage, and they can also become dendritic cells to help elicit an immune response. Macrophage cells can be stored in the spleen as a reserve. Monocytes are responsible for phagocytosis of any foreign substances in the body. They perform this task with the help of intermediary proteins like antibodies and complement through a process called opsonization. They will coat the pathogen and bind to it. If it's a microbe, they'll bind directly to the outside of a microbe through what's called pattern recognition receptors that recognize pathogens. So monocytes are also capable of killing infected host cells, and they'll use that. They'll do that by using antibody-mediated cytotoxicity methods. So these guys are another great way to defend against tissue infections. And once they activate and engulf or degranulate around the infection, they can present part of the foreign invader after it's been digested. They can present these particles to the immune system so the immune system knows what to look for. Okay, let's do the eosinophils next. Eosinophils typically make up about 1 to 4% of the total population of white blood cells, and these appear sort of a brick red color after staining with an eosin stain. These cells have an acidic pH inside the cytosol, which is why they stain this way. And the abundant granules makes them look very bubbly inside. Sometimes under the microscope, they can even look like a raspberry. So these granules take up the red stain and contain a lot of chemical mediators like histamines and proteins like peroxidase, ribonuclease, which will break down foreign RNA, deoxyribonuclease for destruction of foreign DNA, lipases, and several other proteins that act as enzymes to destroy foreign invaders. These chemical mediators get released by a process called degranulation. Once the eosinophil has been activated, the degranulation process will be, the granules will come out of the cell. So the cell basically dies if it encounters a pathogen, but those granules will come in contact with the pathogen and hopefully destroy it. So this process is toxic to both the parasite and the host cells nearby. A mature eosinophil will circulate for 8 to 12 hours and can survive in the tissues for an additional 8 to 12 days. Eosinophils are essential in fighting viral infections and will respond to helminth infestations. Helminthes are a class of roundworms. So roundworm infections, they probably attach to your intestines and they would parasitize the nutrients in your GI tract. We usually deworm our horses and dogs and cats, so this doesn't happen to them. But some species can infect humans as well. Eosinophils are often implicated in allergic reactions, including asthma, pathogenesis, and they're associated with how severe the disease will be. There's a medication, I think, out on the market. You guys might have seen the commercial on TV for preventing eosinophil infiltration in the lungs. It's a newer asthma treatment. Recent studies also show that these cell types are involved in antigen presentation to the T cells. And that's another job that was described when we talked about macrophage. Okay, and then last but not least is the basophil. These are the least common of the granulocytes. They represent 0.01 to 0.3% of the circulating white blood cells. So it's very rare to find a baso on one of your blood smears when you're examining patient blood under the microscope. And their name comes from the fact that these cells are basophilic and tend to stain dark blue with basic dyes and stains. So they have an alkaline pH. These large dark blue granules almost completely obscure the nucleus. So that's one of the telltale characteristics are these very dark blue granules, and you can't even tell there's a nucleus in there. So if you were to skip the staining process, it is possible to see the nucleus, which would look like a two-lobed or bilobed structure in the very center of the cell. These granules contain histamine, which can be released when stimulated, and these white blood cells can also access the tissues through the process of diapedesis. Basophils play a role in allergic reactions and parasitic infections, just like the eosinophils we just discussed earlier. Histamine acts as a vasodilator, which promotes blood flow to tissues and often causes the allergy symptoms that annoy us. Basophils are commonly found in high numbers at the site of ectoparasite infections. And ectoparasites mean things that are on the outside of the body, like they would be found if you had a tick infestation.